This tutorial shows you how to set up any one of the 3 Progenitor Cell Assays or PCAs from Preferred Cell Systems. The family of Progenitor Cell Assay kits developed by Preferred Cell Systems consists of HeLO PCA, HemoFLUOR PCA, and HemoLIGHT PCA. HeLO PCA incorporates an ATP bioluminescence readout. HemoFLUOR PCA uses a fluorescence readout, while HemoLIGHT PCA is an absorbance or colorimetric readout. In other words, we've developed these assays so that you can use any plate reader you have available. Progenitor cell assays are methylcellulose free, instrument-based viability and proliferation assays that quantitatively measure stem and progenitor cell populations, usually detected by MethoCult colony-forming unit assays for umbilical cord blood, bone marrow, or peripheral blood products used for hematopoietic cellular therapy. Therefore, progenitor cell assays from Preferred Cell Systems are the smart, easier, faster, more accurate, and cost-effective alternative to the traditional colony-forming unit assays. The technical manuals for these assays can be downloaded from the respective PCA pages under Cellular Therapy Assays on the Preferred Cell Systems website. Regardless of whether you are using HeLO, HemoFLUOR, or HemoLIGHT PCA, all of these assays have a lot in common. Although each PCA uses a different readout methodology, they all quantitatively measure cell viability, proliferation, and growth. All use a 96-well plate format, allowing high throughput capability for large numbers of samples. All PCAs allow any number of replicate wells per sample to be performed. And all PCAs only need 5 to 7 days of cell culture to complete the assay, compared to 14 days for a CFU assay. And all PCAs are methylcellulose free. They use instead suspension expansion culture technology, making them more accurate, sensitive, and much more reliable. No colonies are produced because all PCAs measure proliferation, which occurs prior to and correlates and predicts differentiation in the CFU assay. All PSAs only require 5,000 cells per well in 0.1 milliliters of total volume. This saves precious cell samples. There are 5 types of MethoCult CFU assays that are used in cell processing labs for cellular therapy products. For each of these assays, Preferred Cell Systems has developed the same 5 types of assay for HeLO, HemoFLUOR, or HemoLIGHT. So PCA1 is equivalent to H4434 Classic. PCA2 is equivalent to H4034 Optimum. PCA3 is equivalent to H4534 Classic. PCA4 is equivalent to H4035 Optimum. And PCA5 is equivalent to H4435 Enriched or MethoCult Express. Whichever MethoCult CFU assay reagent you are presently using, we have the corresponding PCA with either a luminescence, fluorescence, or absorbance readout to give your results a superior edge over the CFU assay. The procedure for setting up a progenitor cell assay is the same regardless of whether you are using HeLO PCA, HemoFLUOR PCA, or HemoLIGHT PCA. For all 3 assays, there are 3 steps. First, prepare the target cell suspension and adjust the cell concentration. Use your own protocol to prepare the cell suspension. Remember, a mononuclear cell fraction or higher is recommended. Adjust the cell concentration to 500,000 cells per milliliter. You can also use 750,000 cells per milliliter. In either case, the cell concentration will eventually be diluted 100-fold down to either 5,000 or 7,500 cells per well. It is very important that the adjusted cell concentration not be based on viability or total cell number, but on the number of mononuclear cells present. You will be adding the cell suspension as 10% of the total culture volume. You will need to dispense 0.1 milliliters of the culture master mix into each of the replicate wells. The number of replicate wells you perform is up to you, but we recommend a minimum of 4 replicate wells per sample. The total culture master volume, that is, the master mix plus the cells, will contain 10% volume of the cell suspension. Next, label the number of tubes equivalent to the number of samples being tested, followed by the addition of the master mix and cells. This will prepare the culture master mix for plating into the wells of the 96-well plate. The cells will then be cultured. The final step will be to measure viability and proliferation ability using either ATP bioluminescence, fluorescence, or absorbance, depending on the assay kit readout you are using. Measurement of each of the readouts will be described in a separate tutorial. Let's now start the procedure. Depending on the MethoCult CFU reagent you have been using, the bottle of master mix from the kit will be for PCA 1, 2, 3, 4 or 5. The bottle of master mix has been thawed and the contents mixed gently. We should be setting up cultures for 2 cell samples, so 2 sterile tubes will be labelled 1 and 2. Next, we've prepared our 2 sample cell suspensions. These will be sample 1 and sample 2. The working cell concentration based on the mononuclear cell count is set at 500,000 cells per milliliter for each sample. This means that the final cell concentration will eventually be 5,000 cells per well. We're going to set up 6 replicate wells per sample. We will now dispense 0.585 milliliters of the master mix into each of the empty tubes. If we only wanted 4 replicates, we would dispense 0.405 milliliters of the master mix. And if we wanted only 2 replicates, we would dispense 0.315 milliliters of the master mix. Once the master mix has been dispensed into each tube, we won't be needing it anymore. Gently mix cell sample 1 to resuspend the cells and transfer 0.065 milliliters into tube 1. This volume represents 10% of the final volume of 0.65 milliliters, which will be sufficient to prepare 6 replicates of 0.1 milliliters each, leaving us approximately 0.05 milliliters left in the tube. With this step, we are diluting the cell suspension 10-fold, so its concentration in the tube is now 50,000 cells. If you were preparing only 4 replicates, the cell volume would be 0.045 milliliters, and for 2 replicates, 0.035 milliliters. Now gently mix the second cell sample and dispense 0.065 milliliters into tube number 2. We will not be needing the 2 tubes containing the cell suspensions, and these can also be removed. The 96-well plate can now be removed from its envelope. It is worth noting that for bioluminescence, white plates are used, for fluorescence, black plates, and for absorbance, clear plates are used. The next part of the procedure involves dispensing 0.1 milliliters into each of the 6 wells of the first and second row of the 96-well plate. These will correspond to sample 1 and sample 2 respectively. This will reduce the cell concentration another 10-fold, so that the final cell concentration per well will be 5,000 cells. If you're using a non-repeater pipette, then for each transfer of the culture master mix to the well, change the pipette tip after each addition. If you're using a repeater pipette, changing the tip will not be necessary. In this case, we will dispense into each well separately, which will require changing the pipette tip after each addition. It is important to dispense the culture master mix into the center of the well, not on the walls of the well. Once the 6 replicate wells for sample 1 have been prepared, repeat the process for sample 2. Replace the lid onto the 96 well plate. Transfer the 96 well plate to a humidified container. This is described in detail in the technical manual. Then transfer this to the humidified incubator set at 37 degrees Celsius with an atmosphere containing 5% carbon dioxide, CO2, and, if available, 5% O2. The low oxygen tension, which is approximately 45 millimeters of mercury and approximately similar to that of venous oxygen tension, will help reduce oxygen toxicity and increase plating efficiency. Incubate the plate for a minimum of 5 days. If the master mix stimulates a stem cell population, the plate can remain in the incubator for 7 days. During this time, there should be a 2 to 3-fold increase in proliferation compared to day 5. This provides increased sensitivity of the assay, but may also increase the coefficients of variation. The first thing you are going to realize when using PCA or any other instrument-based hematopoietic assay from preferred cell systems is that there are no colonies to count. The information you obtain is either in luminescence, fluorescence, or absorbance units, a far cry from looking down a microscope and differentiating colonies. People in the field have been taught, often erroneously, that the only way to determine the function of hematopoietic stem and progenitor cells is to grow colonies. This is very misleading because the primary function of stem and progenitor cells is not to differentiate, but to proliferate. There are 4 important characteristics that need to be emphasized. First, cell proliferation occurs before differentiation and can therefore be measured earlier and quantified. That is why the PCAs take only 5 to 7 days. If cell proliferation occurs, cell differentiation will follow by default, under normal conditions. Therefore, proliferation will predict and correlate with differentiation. This is why there is a direct correlation between Halo PCA, HemoFLUOR PCA, or HemoLIGHT PCA with the CFU assay. Third, when stem and progenitor cells proliferate, they do so exponentially over a period of time. There is no exponential function in differentiation. Therefore, once cells begin to differentiate and mature, there will be no increase in colony formation. Finally, all cell instrument-based assays from preferred cell systems are quantitative. There is no subjectivity and there are no assumptions to be made. What you get are the real results. If calibration and standardization has been performed, which is possible with all Halo assays and can to some extent also be performed with fluorescence and absorbance assays, measurement assurance parameters can be set. These provide you with a clear indication of whether to accept or reject the results. Once these characteristics have been properly considered, it is then easy to understand and interpret the data from PCAs and other instrument-based assays. For a CFU assay, even growth of a single colony is often considered a positive result. There are no acceptance, rejection, or other measurement assurance criteria for a CFU assay to tell you otherwise, because there are no standards or controls to help you. So let's take a look at some results. This graph shows a comparison between results from HeLO PCA and the CFU assay for umbilical cord blood cells. The x-axis shows each of the PCA variations, that is PCA 1 to 5, and the corresponding MethoCult CFU reagents. The left y-axis shows the mean ATP concentration in micromolar, measured in this case on day 7 using 5,000 cells per well. The results represent the mean of 6 replicate wells. The right y-axis shows the total number of colonies counted on day 7 and then again on day 14, also obtained with 5,000 cells. Let's first take a look at the colony counts on day 7 and on day 14 for each of the different CFU formulations tested on umbilical cord blood cells. The most important thing to notice is that regardless of the CFU formulation, the number of colonies counted on day 7 and on day 10 is very similar. This is because once a colony forms, no new colonies are formed with time. The colonies only get larger with time due to the exponential proliferation of the cells that have the ability to proliferate. You will also notice that the number of colonies is greater for the most primitive stem cell population, namely H4453, than any of the other formulations. I should add that our formulation, equivalent to MethoCult H4453 or PCA5, contains thrombopoietin or TPO. This not only potentiates colony formation, but also produces true CFC GEMM colonies. Now let's look at the ATP concentrations produced at day 7 for each of the PCA formulations. First, stem cell populations have greater proliferation ability than progenitor cells. Second, the proliferation ability of all formulations is greater than that of colony formation. This is because colony counting does not take into account the size of the colonies, and even if it did, it would not be able to quantitatively measure cell proliferation. In other words, even though colony formation requires cell proliferation, the CFU assay is not a cell proliferation assay. If you are using HeLO PCA, as we are here, then the dotted line set at 0.04 micromolar of ATP represents the acceptance-rejection level for cell proliferation. No such level exists for the CFU assay. Finally, the vertical lines on top of the colored bars represent the standard deviations, which in turn can provide the percent coefficients of variation or percent CVs. You can expect CVs for ATP assays to be about 15% or less, while for the CFU assay they are significantly greater. This means that the power of the CFU assay to provide statistically meaningful results is extremely low. There are many other advantages to using instrument-based assays for cellular therapy, and these can be found on the Preferred Cell Systems website. This concludes the PCA setup procedure tutorial. Please see the separate tutorials for measuring bioluminescence, fluorescence, or absorbance. The PCAs were designed as an instrument-based, more reliable and consistent alternative to the CFU assay in the cellular therapy laboratory. The PCAs are a stepping stone to the more sophisticated and informative assays designed to predict stem cell quality, optimize procedures for preparing stem cell products, and ultimately measuring stem cell identity, purity, and strength prior to use in patients. We hope that at least one of the PCAs will be your stepping stone to greater rigor and scientific excellence by providing the patients you are helping with high quality and potent stem cell products. If you have any questions or comments, please do not hesitate to contact us. We are here to help you in every way we can so that you can get the most trustworthy and consistent results possible.